Very good morning to all of you. Welcome to this pleasant Monday morning. Yeah, a little bit colder today. Fine. So we are studying the enzymatic pathways, wherein we have already completed the glycolysis process, the intermediate processes, or the ED pathway we have completed. We have completed the TC or the citric acid cycle. Now there is another pathway by which the degradation of sugar takes place. This is called as pentose phosphate pathway, or it is also called as HMP shunt. It is called as HMP shunt. Now let us understand this particular pathway. Now if you see here, the pentose phosphate pathway is also called as the hexose monophosphate pathway, HMP shunt phosphogluconate pathway. So bear in mind, in the examination, it may be asked like explain the hexose monophosphate pathway, or it may be write short note on HMP shunt, or explain in detail the various reaction associated with phosphogluconate pathway. So all these words that I have uttered, all these, they have a very common meaning that all these are nothing but the pentose phosphate pathway. It is an alternative route for the metabolism of glucose. It is more complex pathway than glycolysis. So it is not that much simple because there are a number of reactions that takes place in it, and therefore it is a little bit complex kind of pathway. It is an anabolic reaction in nature. It takes place in the cytosol. Where this pentose phosphate pathway takes place, it takes place inside the cytosol. The tissues such as liver, adipose tissue, adrenaline gland, erythrocytes, testes, lactic mammary glands are highly active in the HMP shunt. So these are various different sites inside the human body where this pathway, the pentose phosphate pathway, acts very correctly. It concerns with the biosynthesis of NADPH and pentoses. Now, what is its biological significance? Importance of NADPH. NADPH is used for the synthesis of certain amino acids involving enzymes glutamate dehydrogenase. So, with the help of this glutamate dehydrogenase, the NADPH is helpful for generation of amino acids, certain amino acids. It is used for the biosynthesis of fatty acids and steroids. The NADPH keeps the glutathione of the RBC in reduced state to preserve the integrity of the RBC membrane. So, this NADPH has its own significance in the human body. It is helpful in various different reactions as well. Moving further. The NADPH keeps the Fe2 ferrous ions in the hemoglobin in the reduced state. The process of phagocytosis is reduced by NADPH. It is required for detoxification of drugs. Now, importance of peptoses or pentoses. What is this pentoses for? The pentoses and its derivative are used for synthesis of nucleic acids, DNA and RNA, and many nucleotides, ATP, NADP plus, and coenzyme. When an organism is growing on pentose sugar, 5 carbon, this pathway is used to produce carbohydrate for certain cell wall synthesis as well. Now, the reactions of the pathway. What are the different reactions? The sequence of reactions of HMP shunt is divided into two phases, oxidative and non-oxidative phase. Oxidative phase, glucose 6-phosphate dehydrogenase is an NADP dependent enzyme that converts glucose 6-phosphate into 6-phosphoglucanolactone. It is hydrolyzed from 6-phosphogluconate. They undergo decarboxylation to form ribulose 5-phosphate. See here, glucose 6-phosphate converted to 6-phosphoglucanolactone, 6 carbon enzyme, glucose 6-phosphate dehydrogenase. This glucose 6-phosphoglucanolactone now gets converted into 6-phosphogluconate, correct, with the help of enzyme hydrolysis. The phosphate glucose dehydrogenase, it undergoes reduction process and it gets converted into ribulose 5-phosphate, again a 6 carbon compound which is there. So this is the oxidative phase. There are various 4 different reactions, so the 1, 2, 3 and 4, but this is all oxidative. It means it works in the presence of oxygen. Now, second phase, that is non-oxidative phase. The enzyme epimerases converts ribulose 5-phosphate into xylulose 5-phosphate. The enzyme ketoisomerases converts ribulose 5-phosphate into ribose 5-phosphate. The enzyme transketolase converts the transfer of two carbon moieties from xylulose 5-phosphate and ribose 5-phosphate to give a glyceraldehyde 3-phosphate and pseudoheptidose 7-phosphate. Transketolase is dependent on the enzyme thymine pyrophosphate TPP and Mg2 plus ions. The overall reaction may be represented as 6 glucose 6-phosphate plus 12 NADP plus 6 H2O gives 6 glucose 6-phosphate plus 12 NADPH plus 12 H and 6 CO2. See, this is the non-oxidative phase where we have left in for ribulose 5-phosphate. Now it can convert into two products. One is xylulose 5-phosphate and it's ribose 5-phosphate. Again, 5 carbon compounds are there. And this xylulose 5-phosphate gets converted glucose 6-phosphate, fructose 6-phosphate, glyceraldehyde 3-phosphate, fructose 6-phosphate, erythrose 6-phosphate, so on and so forth. While the ribulose 5-phosphate goes into glyceraldehyde 3-phosphate into fructose 6-phosphate. So this is the conversion of various different sugars sorry, into different compounds of reduction of the carbon molecule. Regulation. The regulatory enzymes of HMP shunt pathways are glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. The synthesis of both enzymes are induced by insulin. So insulin plays a very triggering role in regulating this particular pathway. The entry of glucose 6-phosphate into PPP, pentose phosphate pathway, is controlled by the cellular concentration of NADPH. So NADPH plays a very vital role in regulating this particular pathway. So the oxidative phase is controlled by NADPH and the non-oxidative phase is controlled by pentosases. So depending upon pentosases and NADPH, this particular regulation of the pathway takes place. Correct? So this is all regarding the pentose phosphate pathway. It is a little bit complex pathway as compared to glycolysis, but varying different reactions in different parts of the body are being regulated by this particular pathway. Correct? So that's all for today. Have a good day.